Hello, welcome back. Let's continue our discussion of the scientific method, and let's focus on the deductive component of the scientific method. Remember what we said. We generated a hypothesis in our previous discussion. In that inductive process, we went from specific observations to our generalized hypothesis. Now, we're sort of going to be reversing things. We're going to take that generalized hypothesis, and we're going to come up with a set of specific predictions. All right, so now we're going from the general to the specific. In essence, what we're saying is, that if my hypothesis is correct, if I assume that it might be correct, then I should be able to do a specific experiment, and I should get results that I can predict fairly accurately if I understand my hypothesis and it has good predictive qualities. All right, it should come out a certain way, and so we want to see if that's the case. Now, what we're doing is we're making the assumption that our hypothesis might be true, and then we're generating our predictions from that. That process of going from that generalized to the specific, or the other way to think of it is the process of making an if-then statement. If my hypothesis is true, then here's what should happen in my experiment. Then we're carrying out that process that's called deduction, or we're doing deductive thinking. Now, we carry out carefully controlled experiments, and we're going to want to talk about these controlled tests or experiments. And what we're actually testing is we're testing the predictions of the hypothesis. You don't want to really think of it as that we're testing the hypothesis directly. We're testing predictions that were generated by the hypothesis. And what we're really trying to do is we're really trying to see if we can prove our hypothesis false. Now, that might seem as a strange way to think about it, but it's actually a very, very powerful tool. It's a very interesting way to approach it, though, isn't it? Because we're saying, here's my idea, and I'm going to carry out an experiment and see if I can falsify it, see if I can prove it to be false. Philosophically speaking, from a scientific perspective, scientists don't really feel as if we ever prove something absolutely true. It's the nature of science to always have that little bit of skepticism, to say that it looks like our idea might be the right explanation, but there's a possibility that it might be incorrect. That's very important, because that means that scientific ideas are always open to be evaluated and tested, and always able to be proven false. That means that if we have a hypothesis, even one we like very much, and we've not been able to prove it false after a repeated number of tests, doesn't mean that the next person that tests that predictions of that hypothesis might not be able to show it to be false. We always want to leave that door open. So that means that's not going to be a search for absolute truth. Instead, we're going to feel as if we're getting closer and closer to the best explanation for something, but we might be wrong. That's why our hypothesis has to be able to be falsified. If it's not able to be falsified, if it doesn't have the potential to be falsified, then that means it's not a good scientific hypothesis. All right? So let's say that I've tested a hypothesis. I don't reject it. I don't prove it to be false. We don't jump to the conclusion and say, oh, well, that means that we've proven our hypothesis to be true. Instead, what we say is that we've provided support for our hypothesis, that our hypothesis seems to be correct. Now, I know that you'll hear people on TV and you'll hear scientists, unfortunately, saying, we've proven this and we've proven that. And I cringe every time I hear it, because they're just being a little bit sloppy in the way in which they're using that word proof. So, using the scientific method, we should never say that we absolutely know for sure that something is a particular way. We should simply say we're highly confident. We always leave a crack open, the potential for us to disprove our ideas. Even if we support a hypothesis, what do? We go back and generate new predictions, and we test those predictions. And even if those are supported, we go back and we do it again. Remember, I mentioned that whole idea of reiteration, that this is a process that we do over and over and over. We want to build confidence. We want to feel confident that the conclusions that we've come to, the results that we get that lead us to conclude that our hypothesis can't be right? Can't be falsified, allows us to feel more confident that our hypothesis is probably correct. Now, if you think about it, what that means is that if we go through this process, we've repeatedly tried to falsify a hypothesis. We've also gone through the process of, doing this with related hypotheses. Hypotheses that are developed from that same set of observations and questions we came up with at the very beginning of this process. And we repeatedly test the predictions of those hypotheses, and we can't disprove those. Think what we're doing. Each time we generate predictions, do an experiment, can't disprove something, that builds our confidence. So what happens to our hypothesis if it goes through this kind of process? If we do this kind of repeated prediction making, testing, evaluation of the ideas of hypothesis or set of hypotheses that are related to each other, then we talk about the hypotheses as moving up to the level of a theory. Now think about what that means. How many times on TV have you heard people say, oh, that's just a theory? Well, from a biologist, from a scientist's point of view, then they clearly don't understand the idea of a theory. A lot of times people in talking in everyday life use the word theory for guess, or just your idea or your opinion. Theory in a scientific sense, though, means something very different. This is a very robust, a very strong, a very tight set of hypotheses that have not been able to be disproven, even after having been tested, their predictions, repeatedly, over and over again. So when we talk about a scientific theory, we're talking about something that we feel very confident about. It doesn't mean that it could be wrong. Remember, we always leave that door open to say that even if I have something that I call a theory, it still might be able to be shown to be false. It just depends upon how we ask questions about it and what kind of predictions we make and what kind of tests we carry out. Scientific theories have been turned over in the past, and a good scientist should be looking for that opportunity to fine-tune their theories, to make them better explanatory tools. Because if I have a set of related hypotheses that come together in a generalized theory, that theory should have a broad explanatory power. It should be able to explain a lot of things that we see in nature. That's what we're looking for, a tool to try to explain nature. And so our goal is to try to develop these theories from our hypotheses that we work with. So please realize that when the word theory is used in science, we're talking about a really strong, well-researched idea, set of hypotheses, an explanation that seems to work in virtually every case that we try it. It doesn't mean just somebody's wild guess or their opinion. So please be careful how you think of that word and use that word theory. I'm going to use it this semester in the scientific sense, and you'll understand that when I use the word theory, I'm meaning something that has this level of confidence associated with it. All right, so now consider how we're going to carry out that experiment that we're going to do. We're going to want to feel confident that we've done it correctly, that we've been careful in how we've carried out our experiment. And a key feature of that is to have a good idea about how we've controlled the characteristics of the experiment and the organisms we've used in the experiment. So the idea of control is very important in the experimentation process. Think about control in the sense of the environment. That's one way that we can consider controls. Of course, we can think of those as being the environmental conditions that, if we're in a laboratory, we can control them fairly well. So we tend to think of laboratory experiments as having a high level of environmental control. Controlling the things like temperature and lighting, humidity, chemical conditions, and of course, laboratory environments allow us to do that. Now, some people might criticize laboratory experiments because they'll say they're not natural. Look at the problem associated with a natural kind of experiment, an experiment done out there in nature somewhere. We tend to have lower control ability. We can't control the environment as well. It doesn't mean that we don't have any control, but field experiments tend to have a lower degree of environmental control, and for some people that makes them, in their minds, seem less valuable. I would argue that both types of experimental environments are what we want to use. Laboratory environments with their high level of control, and natural environmental experiments because they do more closely match the real conditions that living things exist in. Now, a second way we consider this idea of control is the idea of what's called a control group. A group of organisms or living things that we're doing experiments with, where we've controlled that group in terms of the characteristics of the group and the conditions that they're exposed to. Ideally, they should be identical to the experimental group, except for that one key factor that we're going to be testing. Now, sometimes that's very difficult to accomplish, and lots of laboratories spend lots of money to try to make sure that they have groups of organisms that match each other well. Imagine if you're not doing a laboratory experiment and trying to find groups of humans, for example, where we've matched them well, so that we have this control group, and then we have an experimental group, and all the conditions that affect them, or the characteristics of all the members of both those populations, are the same, except for that one key factor that we're testing, that one exception where the experimental group is exposed to something different than the control group. This can be very difficult, but it's very important. It's one of those things you want to consider as you look at experiments. You want to think about, gee, how did they control the environment? What kind of control groups were used in this experimentation? Now, the experimental group, as I mentioned, should have just one key factor that's different than the control group. Not always the case. One of the things that you want to be critical of as you look at the results of experiments, or as you analyze people's experiments, is how well did they achieve this goal? An experimental group that varied from the control group by only one characteristic, because of course we want to compare those two groups. Did that one characteristic cause the experimental group to have a different set of results than the control group? That's why this is so important. All right, now, this isn't the only way in which experiments can be done. Sometimes nature carries out the experiment for us, and we have to use the comparative method. A flood comes through an area and washes down through a valley, and we compare the side of the valley that got hit by the flood with the side of the valley that didn't get hit by the flood. Well, that's a naturalistic experiment. That's not one that I carried out. I don't have a high degree of control over it, but I can still analyze it and compare those two regions and try to determine what were the factors that influenced the change in one environment versus another. So this comparative process, while it lacks a certain degree of control, can still be valuable, and it's used quite a bit by scientists, especially those that are studying natural conditions out in the environment. It can be difficult, however, but it's a very important technique that can be utilized. Well, what we looked at here is the second component, or the focused on the second half of the scientific method, from the hypothesis on, the deductive process, the development of those predictions that could be used for experimentation. And one of the key ideas I try to get across to you is this idea of the greater the degree of our control, or the understanding we have of the variables in our experiment, the better able we are to determine if our results are appropriate. And finally, I mentioned to you that idea of the comparative method, which is a naturalistic experiment, not one that we intentionally carried out, but in a sense one that nature did for us. And we're looking at the results of it and trying to understand what are the factors that influence the changes or the differences that we see. Well, I hope this helps you understand the scientific method better. And in particular, I want you to consider what I said about the idea of the word theory. And think about how many times you hear it. And every time you hear it, question whether the person is using theory in the correct scientific sense. Well, thanks very much. I look forward to talking to you next time.